I am Pradipta, and today I'm going to talk to you about a work done at the School of Computer Science, Carnegie Mellon, about a feature-based discriminative method for motif search in eukaryotic genomes. And this is joint work with my advisor, Eric Xing, and my coworker, Wenjie. So the basic problem here is to figure out where transcription factors, which are a kind of protein, bind to the DNA to either aid or to repress protein transcription. Learning such patterns, which might be indicative of these binding sites, and predicting putative binding patterns has been an open problem for over 20 years now, but we still do not perform as well as we would like. Binding sites in higher organisms typically occur in clusters called cis-regulatory modules, and they are noisy copies of each other, like this. So typically, the segments marked in red would be binding sites, and you can see that they're pretty similar to each other, and they occur in spatial contiguity on the DNA, close to the transcription start site. The basic organization of the talk would be as follows. We would start off with the motivation for our problem, move on to the schema of how we solve it, look at some empirical analysis, then take a look at the results, and go on to conclusion. So without further ado, moving on to the computational formulation, what we want to do is that given a sequence of nucleotides, we would like to figure out exactly where the transcription factor may be binding. And there are basically two approaches when you have a large amount of data available to you. One would be unsupervised methods for finding, and one would be supervised methods. And what differentiates these are the fact that for supervised methods, you have some training binding site data available, but for unsupervised methods, you don't. And we will be looking at supervised methods today, so we will take it from here. So supervised binding site prediction is traditionally been done by modeling the binding sites as fixed with tables of position-specific frequencies, like this. So typically, for every position in the binding site, we have a distribution over the nucleotides, which would model the binding site itself. Each such instance you would find in the regulatory region would be called a motif. And the table itself is called a position weight matrix. So traditionally, when you wanted to find binding sites, what was typically done was to scan a fixed length window across the genome and compare the binding site model with a background model. And in cases where the scores for the binding site model was much higher, we would claim that it would be a binding site. So what is the problem with this approach? Problem is that since binding sites are noisy copies of each other, you would run into a lot of false positives, as you can see here. Other than the actual binding sites, the method would pick up all these wrong false positives. So traditionally, to overcome this problem, what has been done is to go on to more complicated generative models, such as hidden Markov models. What these do is have a statistical formalism in place, which would allow for generating each sequence of the nucleotide based on an underlying model, which somehow models the locations of the binding site. So typically here you would have two kinds of states, motif states and background states, and they would be simulating characters from the corresponding PWM or the background distribution. And then, given some training data, you would learn parameters for this model, and accordingly, you would try to find new binding sites using these parameters. However, there is a small problem with generative models, which we have found that generative models may tune to the noise instead of signal for noisy data, because what they do is they model the entire joint probability of both the sequence data and the labels itself. But what seems to be a bigger problem these days is, especially in the past 3 or 4 years, is that these generative models, their performance has saturated to some degree. Sophisticated models like BASIS, which was actually a work done in our group, which actually uses hierarchical, generalized hidden Markov models, which models arbitrary distributions over distances, as well as a hierarchical framework, have not significantly improved the state of the art. The other problem which we find with HMMs is that it's difficult to include non-sequence information for binding site evidence into the model. And over the last 3 or 4 years, there have been a bunch of work done to try to incorporate various kinds of evidences to figure out where binding sites may lie. The most traditional among these are the phylogenetic motif finding methods. What they typically do is they look at the neutral theory of Kimura and say that functional regions, such as binding sites, should be more conserved than non-functional regions. And this started with the simple but very influential Lutz Vista paper in 2002. And there has been a bunch of work on this, including work in our group. Other than this, people have tried to include distance from transcription start sites, distance between transcription factor binding sites, epigenetic data such as nucleosome affinity, as well as trying to somehow combine all these features together. The model we propose is to try and combine diverse sources of evidence very neatly into one framework. And we use a very powerful statistical integrational device called conditional random fields. So what is the advantage of this method? How is this better than other methods in the field out there? Firstly, since it's a conditional random field, it is a discriminative model. And by discriminative model, what we mean is that it maximizes the conditional probability of the label given the sequence rather than the joint probability. And as a result, it is less sensitive to noise in the data. Further, what we have done is we have chosen very carefully a set of features. By features, we mean any kind of evidence that can help predict binding sites. We will look at what kind of evidences these might be, like phylogeny and other stuff, in more detail shortly. We have chosen carefully from the literature. So moving on to the schema, what we typically do is for the estimation part of our model, the input is a set of sequences which have corresponding feature values for each nucleotide and positions of the transcription factor binding site. And what we get as output during the estimation or training phase is the learned conditional probability model, which basically are feature weights with each weight corresponding to one feature. In the inference, what we take as input is a set of sequences for which we do not know where the binding sites lie. And we take the learned model from the training phase, and we get as output not maximum likelihood, but maximal a priori labels corresponding to a nucleotide specifying its state, whether it's a binding site or a background, and so on. So this is the main schema of our diagram. It looks complicated, but we will go over this piece by piece. So let's first look at what we do. We take some regulatory DNA sequences, some training binding site data, and we perform some empirical analysis by which we choose a set of features which might be feasible for that data set. Then, for those feature sets, we compute the scores for each of the features. So the features might be phylogeny-based features, such as presence or absence of conservation. They may be binding specificity-based features, like PWM scores. They may be chromatin stability and accessibility features, such as nucleosome binding affinity. They might be transcriptional features, such as proximity to transcription start sites. And you could look them up in databases, or if you are collaborating or you are a wet lab biologist, then you could directly do the biological experiments yourself to generate those data. Then what we do is taking those feature scores and the training binding site data, we estimate the parameters of the model, basically the feature weights. And we then further, using the feature weights, we would perform inference to find out new binding sites. And by testing them biologically, we can add to the training binding site data and thus close the loop. The estimation that we perform can be converted to a convex optimization problem, which makes life pretty easy for us. What we do is we use a quasi-Newton method such that we can do some kind of a gradient-based approach. For the inference, this conditional random field, at least for the kinds of conditional random fields we use, have corresponding analogs of the HMM inference algorithms. The nice thing about this means that we can add certain kinds of features and still expect to use the same inference algorithm. With generative models, what happens is when you try to incorporate new features, you end up with inference problems so difficult that you might have to go for approximate methods, which would kind of give you bad results. Let us now look at the exact features that we employ and the empirical analysis that we did. So one kind of feature are sequence-specificity features, such as the PWM-based score for a particular subsequence. Self-complementarity, how similar is the binding site to its reverse complement. This could be used for binding sites where the PWMs are pretty self-similar in the reverse complement. We try to model the cis-regulatory grammar by modeling transition probabilities between states of the model. By states, we mean background, binding site, CRM backgrounds, and so on. And along with the PWM score, these are the two main sources of evidence which are traditionally used in hidden Markov models. But we go on to add many more such features. One such feature are evolutionary data. Conservation, as I told, that function implies conservation. So some kind of indicator of whether the sites are conserved. Presence of repeats. Repeats, whether they are duplicated binding sites or whether there is functional turnover there. Chromatin stability-based features, we kind of have tracks or features which calculate the GC content, which would give a good indicator of chromatin stability as well as the melting temperature, CPG islands, nucleosome binding affinities, because nucleosomes affect DNA wrapping and therefore kind of affect the accessibility of transcription factors to the DNA. Distance and location-based features, such as distance to the transcription start site, presence in the 5' or 3' UTR or the intron. What we did after training our model was to take a look at what the feature weights and the average values of the features were. This can be very illuminative, as it can help us understand what kind of features correlate well with binding sites. For example, we looked at GC content for our data set in Drosophila, and we checked that for background, motif, and for CRM non-motif regions, we actually got reasonably discriminative scores. Similarly, fastcons is an evolutionary conservation score using the fastcons tool. And we found, as expected, that motifs have a higher conservation there. Sometimes it is even more illuminative to actually analyze the distribution of these scores rather than just the means. For example, for the fastcon score, if you bin it, you find that the tails of the distribution are actually more discriminative than the middle part of the distribution. Finally, what we did was we checked that even if features are discriminative, they might not really meld very well into the CRF framework, or sometimes there might be redundancy involved. So whether the features that we add are actually discriminative in the context of the model, we checked by cumulative probabilities of both true positives that we predicted and false positives that we predicted by incrementally adding one feature after another. And basically, we wanted to see whether the true positive line and the false positive line had a gap. Moving on to the results. We performed simulation based on a different model, a generative model, HMM-based, for a sanity check. And we find that our model, even though it's not the best, it still does reasonably well. For the actual biological data, we used Drosophila data for early developmental CRMs. We had 35 sequences and close to 600 binding sites. The cis regulatory modules are marked out in blue here, and the binding sites in red. We did leave one out cross-validation, which basically means that we trained on all but one of the sequences, and we tested on the remaining one. And we calculated precision and recall, where precision is number of true positives upon true positive plus false positive, and recall is number of true positives upon true positive plus false negative. F1 scores were also calculated, which is kind of a single measure for measuring effectiveness, the harmonic mean of precision and recall. We compared our method with a bunch of other state-of-the-art methods. These include AHAB, which is a window-based approach, Cluster Buster, Sister, which are HMM-based approach, BASIS, which is our hierarchical HMM-based approach, as well as two other programs, MSCAN and STUB. And what we found was our precision recall curve performs significantly better than the others. And if you take the default configuration, then our F1 score is also significantly better. It's actually 20% better than any of the other scores. The reason for this, we believe, is the fact that most of the programs that we looked at, especially in their default modes, seem to strike either a very high precision, low recall kind of scenario, or a low precision, high recall kind of scenario. For example, here you would find Sister having a very high recall and low precision, whereas you would find MSCAN having a very high precision and low recall, whereas our program, the CRF, the Discover, actually has a very well-balanced precision recall, which is why it gets very good F1 scores. So here we actually visualize Discover predictions. You can see that the red ones are basically true positives, and the green ones are false positives. And recall that I said a little while back that the main advantage, one of the main advantages of using a discriminative model over a generative model is that it would be less sensitive to noise and would make less false positive errors. If we compare with Cluster Buster, which is state-of-the-art HMM generative model, you'll see that Cluster Buster gets a lot more true positives, but a lot more false positives. So it has so many false positives. So if these predictions were to be used in a bio lab, then it would be difficult to know where exactly to start. So we move on to the conclusion. Future work for us would entail adding additional features like intermotive distances to figure out composite motifs, and other additional features like other epigenetic features. Analysis of assigned weights to different features would be something to go more deeper into to figure out what might be correlating to binding site data, as well as trying to move to an unsupervised motif detection, which is actually a very difficult in the CRF framework, but we are working on it. So finally, the take-home slide. So far, we have seen lots of generative models like HMMs. Problems with these are performance is saturating, the risk of tuning to noise on noisy data, and it is unclear how to incorporate diverse evidence, because it's not clear how to generate those evidences in a generative model. For Discover, our model, it is a discriminative model which maximizes the conditional probability of the labels given the sequence. It is very easy to integrate any kind of evidence into the prediction scheme. while still maintaining the same abstractized estimation and inference techniques. And most importantly, we have our software up on the web. We already have 2 or 3 promising collaborations. If any of you would be interested in a collaboration, we would be more than happy to start one. Finally, the acknowledgement slides. I'd like to acknowledge my advisors, Eric Shing and Veronica Hinman, my co-workers, the travel grant that I got here. This research was funded by an NSF Career Award and a Sloan Fellowship to my advisor, Eric Shing. Thank you. So I can take questions.
I guess the microphone on?
Yep.
OK. There's 2 reasons to build models like these. And one of them is to find new transcription factor binding sites. And the other one is to try to figure out what the cell is using to identify transcription factor binding sites. And I think the thing that distinguishes the 2 models is using evolutionary conservation. So I'm wondering, I mean, I think it's kind of a pity to include evolutionary conservation because it's a bit of a crutch. So are you also interested in the second question as well as the first question?
We are interested in both questions. This paper was primarily built as a prediction paper. So basically, we tried to include evolutionary information in there as well. If we want to figure out what non-evolutionary characteristics might correlate very well with binding site data, we could very easily leave those tracks out and do an analysis based on just that. So it is really...
I think that would be a very valuable piece of work in addition to what you've already done.
Yeah, so I think one of our future... so one of our future work is exactly that. Okay. So we are looking at that, and we are hoping to get a paper soon.
OK, good luck.
Thanks.
Hi, so I have a question regarding your learning principle. As far as I understand it correctly, you use maximum conditional likelihood, is correct?
Yes.
So is there a problem that the parameters overfit to the data? Because normally you take something like a prior to avoid something like this.
Exactly. So we do actually use priors. We have used Gaussian priors. And we actually also used a regularization term. We actually didn't go into all the details because of time. But if you look at our paper, especially in the additional part of the paper, you would be able to find all the mathematical details there. So to avoid the risk of overfitting, we do have a prior, and we do regularization. Yes?
Thanks for the talk. How confident are you in your validation data? Could it be that the false positives, as you call them, predicted might actually be true positives?
That is a problem with binding site data in general, right? So that is exactly one reason why we just use, let me see where it is. That we try to use true positives, but we also might have false positives which are kind of not false positives, but they are actual data which is not yet annotated, right? What we have done is we have collected a wide range of data from different sources in the literature and from several databases like Oregano and the Singapore regulatory database. And we have made as comprehensive a data set as possible. So what might be false positives might actually be interesting to see whether they are true positives.
Your model is actually producing a probability, right? So
yes. So you have a belief.
Yes. Have you considered using an ROC curve to measure?
Definitely.
The performance
so
and
actually,
the area under ROC?
Yeah. So the only problem with an ROC curve is the moment you get true negatives in there, which is what you would have in an ROC curve, the large number of true negatives kind of tend to bias your results. But we have used ROC curves, and we have tried to sort our predictions based on our belief. Any other questions? Okay, so I think we have